Now I request ENEST 157 to start the presentation. ENEST 157.
Good evening to all my dignitaries present, and I am Ujma Mulla, studying in the third year, Bachelor Pharmacy. And the topic which I have chosen for my poster presentation of a review is Omic. Okay, Omic is like the surface. Okay, like for getting the genomics, proteomics. So overall, it's been stated as the Omic, and the title I have given is a global innovative science. The reason behind giving this is that it is like totally innovative, and it is Omic is acting like a global tool to serve the interactions and the study between the drugs and the biological functional molecules. Let me start with the introduction. Um, what is omics? It's like totally, basically, what you can say is the study of an entire set of entities that are present in a class. It has become an efficient tool for the global study of interaction between the drugs and the biologically functional molecules. Like there were different methods that were used for getting to study the interaction between the drugs and the functional molecules. They were consuming more of the time and it was not efficient. So this omic technology is said to be an innovative one, which is totally based on the microarray and 2D gel producing large scales, okay, and which allows the exploration of genome, transcriptomes, and proteomics with greater sensitivity and resolution. Through this, I would like to tell about the objectives that are like omic technology are primarily aimed at the universal detection. Like by having this technology, we can detect the presence of genomics, mRNA, that is transcriptomes and proteins, and the metabolites in a specific biological sample that is being tested. And the different branches of the omics are genomics, transcriptomics, proteomics, ionomics, metabolomics, and phenomics. The first one, that is genomics, it's all about the DNA sequencing with the structural and the functional analysis of the genome that is present in a function. The second one is the transcriptomics, that is the transcriptional regulation of the mRNA. The third one is the proteomics. It is about in a sample about the identification, quantification, and the post-translational modification of the protein. And the fourth one I could say is metabolomics. It's like study of the metabolic profiles having in the different diseases. Like we can study about the different metabolites that are being present with the intermediates, hormones, and other signaling molecules. The fifth one I could say is ionomics. It is about the elements that are present in a biological sample, about giving a total profile, the amount which is being present of the elements, regulation, and the interaction. And then I would say about the phenomics. It's like totally upon the evaluation of the morphological, biochemical, and the physical traits between the genetics and the environmental factors. And here I would tell about the various omic technologies and their role in the biological system. Like what biological system, the role they are playing in is that, as I have stated, the branches of the omics are phenomics, and there it comes is cytomas, genomics, transcriptomes, proteomics, metabolites, ionomics, and metabolomics. So the phenomics and the cytomas are mostly in the cells. The genomics in the chromosomes, transcriptomes are seen in the and in the DNA of RNA. Then proteomics is in the DNA. Then metabolics, proteins, and the ionics and metabolics are seen in the ions. Together, all over these branches of ionomics give us the omic data. That is the amount that is being present and the molecular functional study that is present in the biological sample. Here the omic data gives us the two things. The first one is the molecular network. The second one is internet databases, which is then gives us sets a biomarker, okay, which gets targeted to get the toxicity diagnosis, the mechanism of the toxicity that is being seen in the biological sample, which then is overall seen in the system biology. which gives us overall the bioinformatics about this sample. The third thing that I want to explain is the strategies of the drug target discovery based on the omics. There are 6 targets. The first one is the disease model. Just to study about the disease model, we need to do few of the clinical samples. We need to get the cell model, the animals model, and the disease database. Um, and then overall, when you get on to the omics, there is about genomics, transcriptomics, metabolomics. We have to study and the lipidomic profiles just to get it over. Then the third one is the bioinformatics. The bioinformatics sets us the biomarker which is like naturally present, which gives us the discovery for that biomarker which is being used for that specific omic. Okay, and overall the candidate drug target databases keep to the data mining and redocking. The third one is the fourth one is the functional analysis. We get to know the different functions that are getting into interactions with the drugs. Uh, and then I would say about the there is like pathway analysis of the we can get into RNA analysis, and the fourth one. Here I would explain about the applications that are being used in the pharmaceutical research in the recent way. First of all, just to get into the pharmaceutical research, we have the four things. The first thing is that we have to get the pathway for the way the drug is being administered, so that we can target a drug at a disease. Like different diseases are there, so different pathways are required for different diseases for the drug target therapies. And the first one is, as I said, there is a need of a system biology which gives us the interactions of the interoptics or the bioinformatics, which is then reversible to the network biology inside it. The third one we could say about the pathway and the mechanism, how the drug is acting to the targeted places. Like we can get the target, how the drug is being targeted at the specific position, and there we can get into the drug development. We can get into like this is a drug that will be developed for this target. It will target at this thing. And the last this would be like we can assess the drugment. And the first one is the last one is the pharmaceutical. And the conclusion from my poster would be like the Omix technology has increased sustainably the number of genes that can be detected. From the Omix technology, we can get the number of genes that can be detected simultaneously have the potential to relate the complex mixtures to complex effect. Like different complex mixtures can give a different complex effect in the form of gene expression profiles in the biological sampling. Advances in the Omix technology have the potential to revolutionize our approach to the disease diagnosis. As we use the omic technology, we could get the different profiles of the omics that are being present, and the development of novel therapeutics. Genome based on research has preceded the development of both. The first one is the conceptual framework of the disease understanding. Like through this omic technology, we can come to know the framework of the disease, that is the how is the drug that is getting targeted, how is the drug being developed, its assessment can be done, and the different numbers that are present, the amount is present in a different disease. And the second one is effective tools that can exploit the vast amount of data retrieval from those efforts. And the last one will be the omic is a progressive and useful concept in the biological and the clinical practice. And here I would like to conclude my poster.
Okay, now I request evaluators to start evaluation.
Okay, student, just tell me why omics is important.
Oming would be, I could say it would be important because the olden, like whatever we had, the different way of studying the interaction between the drugs and the biological function was like time consuming. It wasn't that sensitivity to give us the result of the exact number of the drug targeted at that specific point.
And how it is beneficial to detect the human diseases.
So it would be as I said, sir, there are different profiles of the Omix, such as genomics, proteomics, metabolites, like different number of the amount that is present in the biological sample of the below, like such as genomics, proteomics. This can help us, like this amount is like having the such disease, so we can help to differentiate with that, with the biological the system.
Okay, my questions are over.
So it's a good presentation. Omix, can you define in a few words?
So what?
Yeah.
Hello.
Yeah.
So I'm able to hear.
Can you define Omix?
So yeah, Omix is we can say it's the study of the set of entities present in a class.
Because in introduction you have given, it is not clear.
So yes, due to some of my the poster, like I'm so uninformed that.
Okay. Okay. Right. Rest of everything is okay. Fine.
Nothing particular from my side.